Hi everyone, how are you today? My name is Bertie. As some of you may know that I am currently studying psychology at university. One of my ways to study is to record my own voice, then I will listen to it over and over again. Today, I'm going to share with you a psychology subject that I will have to learn for my exam. It's about development, degeneration, and recovery in the nervous system. Let's get started. I will share with you my screen. Biological psychology, development, degeneration, and recovery in the nervous system. Development. Fertilized egg undergoes a period of cell proliferation where repeated cell division results in cell numbers in the embryo. 3 weeks later, gastrulation phase happens. And in this gastrulation phase, a layer of cells produces the nervous system, or the ectoderm. The combination of differential gene expression and cell-to-cell interactions results in neural induction. A region of ectoderm transforms into neural plate. This is the first sign that a nervous system will be formed. What happens next? Let's see. Then after the embryo enters the neurulation phase, what is neurulation phase? It is the formation of the embryonic neural plate and its transformation into the neural tube. The groove deepens until the top edges come together to fuse and form the neural tube, a hollow tube-like structure running the length of the vertebrate embryo that forms the brain and spinal cord. As you can see here in the picture, there is a neural plate on the top, a neural plate border. Then you can see that there is a neural groove there. The neural groove deepens until the top edges come together to fuse and form the neural tube. You can see the neural tube in the bottom part of the picture. Week 4 is the end of neurulation. The embryonic regions will have been established. There are two things happening at this stage. The posterior part of the neural tube forms the spinal cord, while the anterior part of the neural tube forms the brain. Posterior means posterus from Latin. It means coming after or the back of something. Anterior means ante in Latin, which means before or the front of something. That means the posterior part of the neural tube or the back of the neural tube forms the spinal cord, while the anterior part of the neural tube, the front of the neural tube, forms the brain. You can see here in the picture, this is the neural tube here. Forming the foetal brain. What happens at the end of neurulation? At the end of neurulation, the anterior end of the embryo has vesicles. Each of these 5 vesicles form a predictable region of the brain. The stem cells continue to proliferate, which means dividing. Cells form more specialized and neuron-like cells. Determine what type of neuron is produced, including neurotransmitters. What does proliferation mean? It means rapid reproduction of cells, parts, and organisms. You can see here a very important picture, the lateral view here. A here, 3 to 4 week embryo. There are 3 primary brain vesicles: prosencephalon or forebrain, mesencephalon or midbrain, and rhombesencephalon or the hindbrain. Prosencephalon or the forebrain is divided into telencephalon and diencephalon. Mesencephalon or the midbrain still becomes mesencephalon. Rhombencephalon or the hindbrain is divided into metencephalon and myelencephalon. 5-week embryo. You can see here the picture. In 5 weeks, there are 5 secondary brain vesicles. The telencephalon becomes the cerebrum. And then the diencephalon becomes the eye cup, and then the thalamus, hypothalamus, and epithalamus. The mesencephalon becomes the midbrain. The metencephalon becomes the pons and cerebellum, or it's divided into pons and cerebellum. And then the myelencephalon is developed to medulla oblongata. Here is the lateral view. Migration and differentiation. Neurons that can differentiate to form one or more kind of cells, progenitor cells, are produced at ventricular zone. The neurons must travel to their destination to group together with other same types of neurons. You can see here in the picture layers from inside out. Younger neurons migrate through layers of older neurons. You can see here marginal zone, cortical plate differentiation, intermediate zone migration, and ventricular zone proliferation. The newly produced neurons in ventricular zone, their birthplace, travel to the outer surface and destination via radial glia or specialized glia cells. How do they migrate? Well, the process is called inside-out layering process. The first immature neurons migrate the shortest distance from the ventricular zone. The produced immature neurons migrate further. You can see here again the same picture from the last slide as well, layers from inside out. The younger neurons migrate through layers of older neurons. What disrupts the migration and affects brain development? There are 2 things here: genetic mutations and exposure of the fetus to chemical substances. Have you heard about teratogen? You can see here that teratogen is an environmental substance that disrupts normal development. Teratogen can be about alcohol and prescription drugs. Okay, let's see. Okay, we're on the right slide. Connection formation, axon guidance. Neurons that form a circuit have their cell bodies some distance away from each other. At this developmental time, the fully differentiated neurons have axons, which are long extensions from their cell bodies. Axons enable an individual cell to communicate with other cells some distance away by using regenerative action potentials. This process involves the growing tip of axon, called the growth cone. You can see here in the picture that there is a cell body here in the green color, and axon is here like a line here, and then in the end there is a growth cone. Growth cone is the process involves the growing tip of axon. Connection formation, axon guidance. What is axon guidance and the growth cone? Axon guidance is an important step in neural development. It allows growing axons to reach specific destinations and ultimately form the complex neuronal networks throughout the body. Although many aspects of this mechanism remains unclear, it is well established that a dynamic and highly motile actin-based structure found at the growing end of a developing axon, known as growth cone, facilitates this process. Connection formation, synaptogenesis. What is synaptogenesis? It is the formation of synapses. What are synapses? Synapses are the points of contact where information is transmitted between neurons. This is integral for creating brain networks and for the overall architecture of brain connectivity. At least half of human brain tissue is made from gangly branches of nerve cells called neurons. Neurons use electrical signals to communicate with other neurons and send information throughout the brain and body. There is a small space at the point where two neurons almost connect to each other, called a synapse. It is at these spaces where neurons communicate with other neurons by using chemical signaling molecules called neurotransmitters. You can see here in the picture an illustrated image showing the structure of how two neurons connect to each other. The soma here, see the black point there in the green form, the synapse, dendrites, and axon terminals are labeled. You can see that here's the axon and the synapse. You see here the synapse is the points of contact where is it? Synapse. Okay, so here is a space. A small space at the point where two neurons almost connect to each other. It's called the synapse here. A small space. Connection formation. Synaptogenesis. What is synaptopathy? It is dysfunction in synapse function. Synaptopathy examples are ADHD and autism. Neurochemistry. It is the study of the identities, structures, and functions of compounds, neurochemicals, that are generated by and that modulate the nervous system. Neurochemicals include oxytocin, serotonin, dopamine, and other neurotransmitters, and neurotransmitter regulating substances. The neural death and synaptic modeling. Apoptosis, necrosis, and refinement. What is apoptosis? Apoptosis is a programmed cell death in which cells self-destruct to remove unwanted cells. I would remember it as automatic. That means the cell is self-destructs automatically. Necrosis, cell death due to damage or trauma to cells. Refinement, non-active synaptic connections are lost during development. You can see here on the picture on the left-hand side, necrosis. Number 1, always pathological. Cell enlargement, loss of membrane integrity, and then the leakage of content, inflammation, nuclear degeneration. As opposed to apoptosis. Apoptosis, number 1 here, is the elimination of unwanted cells, programmed cell death. Then the cell shrinks, nuclear fragmentation, apoptotic bodies, phagocytosis of apoptotic bodies, no inflammation. Apoptosis is automatic, it's a programmed cell death in which cells self-destruct to remove unwanted cells, while necrosis is the cell death due to damage or trauma to cells. Necrosis versus apoptosis. There are 6 major differences in necrosis and apoptosis. Cell death reasons, necrosis is always pathological, which means cell death happens because of unfavorable stress situations, infections, excessive heat, radiation, or lack of blood supply. Apoptosis is most commonly physiological. It is programmed cell death, which eliminates unwanted cells from the body. Sometimes apoptosis happens in cells with damaged DNA. Cell size. Cell size increases in necrosis and cell shrinks in apoptosis. Nucleus. In necrosis, nucleus undergo pyknosis, fragmentation, and cardiolysis. Apoptotic nucleus breaks into nucleosome-sized fragments. Plasma membrane. There is loss of plasma membrane integrity in necrosis, but plasma membrane remains intact in apoptosis. Cellular contents. In necrosis, there is leakage of cellular contents. Adjacent inflammation. Because there is plasma membrane rupture, there is leakage of content which attracts inflammatory cells. So there is inflammation in necrosis. Apoptotic bodies are phagocytosed, so there is no cellular material to induce inflammation. Hence, there is no inflammation in apoptosis. Critical periods. Specific stages in development when systems and behaviors are shaped for life. Myelination. Myelination is the process by which brain oligodendrocytes produce layers of myelin that wrap around the neural axons and act as a layer of insulation for the transmission of electric action potentials down the neuronal axon. The formation of a myelin sheath a sleeve or sheath that wraps around each nerve cell, neurons. It's a protective layer of fat or lipids and protein that coats the main body section of a neuron called the axon. Myelin is lipid-rich material that surrounds nerve cell axons, the nervous system's wires, to insulate them and increase the rate at which electrical impulses, called action potentials, are passed along the axon. You can see on the picture here on the left-hand side, oligodendrocyte progenitor or PC, and then pre-myelinating oligodendrocyte, as well as myelinating oligodendrocyte. On the right-hand side, you can see the picture of myelin. For example, here you can see the myelin sheath. It's a sleeve or a sheath that wraps around each nerve cell. You can see here the node of Ranvier, the Schwann cell, which is the orange color there, and the axon terminal. You see also the nucleus, which is the green thing there, the cell body, and dendrite, which is like a tree. It looks like a tree, dendrite. Synaptic plasticity. Change that occurs at synapses, the junctions between neurons that allow them to communicate. The idea that synapses could change and that this change depended on how active or inactive they were was first proposed in 1949 by Canadian psychologist Donald Hebb. How do synapses change their strength? After a neuron fires an action potential, there are three main steps to synaptic transmission. As seen in the diagram below, synaptic plasticity can change either 1, the amount of neurotransmitter released here, or 2, the number of postsynaptic receptors available. Both have the effect of altering how much electrical current flows through the ion channels 3. In other words, these processes change synaptic strength. You can see here neurotransmitter release and then binding of neurotransmitter to postsynaptic receptors, and then opening of ion channels in the postsynaptic neuron, which allows electrical currents to flow in or out of the cell. On the right-hand side, you see a picture on synaptic plasticity that can change either the amount of neurotransmitter released, 1, or the number of the postsynaptic receptors available, resulting in changes in synaptic strength. You can see here are the neurotransmitter with the synaptic cleft, and here the spine, and then the dendrite. This is normal, and then potentiated here. But there are also disadvantages of synaptic plasticity. Allows for inappropriate connections to be made, which means unwanted memories or behaviors emerge. Intense experience causes reduced ability to form memories. For example, soldiers didn't remember the time period around the time of the injury. Those are the disadvantages of synaptic plasticity. Sex differences in brain development. Brain development is influenced by sex hormones exposed during the development. The default pattern is to produce a female brain, Masculinization requires exposure to testosterone, produced in the male fetus when the testes develop, which is determined in the presence of genes on the Y chromosome. In female, the testis development absence results in the ovaries development and other hormones related to female reproduction. For example, the estrogens. Male and female brain structures differ in many ways. The size of the individual neurons, the number of neurons in a brain nucleus, and the size or weight of the whole brain. Degeneration involves the loss of synapses without significant reduction in neural cell numbers. Synaptic loss, reduced cognitive power, and then it's seen in normal aging. Pathological loss, Pathological loss, neuronal populations loss through injury or pathological process, results in loss of functions. Neuronal death may occur in healthy neurons if basic requirements of the cells are not met, or it also involves intracellular pathological process. Let's talk about the healthy neurons, the neuronal death that happens in healthy neurons if basic requirements of the cells are not met. For example, the stroke patients, disruption of blood supply to neurons, reduction of oxygen and glucose, and waste removal result in cell death. Smaller scale neuronal death, for example, TIAS, transient ischemic attack. Small region has diminished blood supply for a short time, repeatedly may result in gradual decline to the brain region affected. People may be more aggressive or pessimistic. With regards to intracellular pathological process, it's similar to apoptosis. In response to genetic mutation or environmental insult, or both of them, that cause the neuronal death. Huntington's disease. The cause is the genetic mutation. It comes from the UK. Is monogenetic. The protein produced by the mutated gene or huntingtin, critical for all normal cells, including brain neuronal activity. The mutation happens in the region of the genetic sequence that is repeated more frequently than in the normal version of the gene. On the picture here, you can see the Huntington disease brain and the normal brain. Huntington disease brain. There is a difference here than the normal brain. You can see here in the picture. Repair and recovery. Neurogenesis. What is neurogenesis? Neurogenesis is the process by which new neurons are formed in the brain. Neurogenesis is crucial when an embryo is developing, but also continues in certain brain regions after birth and throughout our lifespan. Let's look at the picture here. Neural stem cells have the potential to generate all neural cell types. They differentiate into neuronal progenitor cells, which give rise to neuron, or glial progenitors, which give rise to glial cells. You can see here the neural stem cell, proneural genes, and then Gliogenetic signals from neural genes. They are divided into neuronal progenitor and glial progenitors. Here is the neuron, astrocytes, and oligodendrocytes. Neurogenesis, gliogenesis. Neurogenesis. Let's go back here. The neurogenic capacity restricted to certain populations of neurons. Neurons of the olfactory or smell, born continuously throughout life within the subventricular zone, the lining of the lateral ventricle. Neurons in the temporal lobe called the hippocampus. That means this neurogenic capacity has two parts here. Neurons of the olfactory, smell, and neurons in the temporal lobe called the hippocampus. What is immunohistochemistry? It is a technique for the visualization of proteins in tissue. A technique for the visualization of proteins in tissue. Immunohistochemistry. Immunohistochemistry. We should remember that. Behavioral genetics. Genome. What is genome? It's the complete set of genes in an organism. Gene. Gene is the part of chromosome that is the blueprint for a protein. Allele. Alternative form of a gene. Polymorphism. Multiple types of allele or control region. Genotype. The complete set of alleles an individual has. It determines characteristics and traits of an organism. Phenotype. Physical or biochemical characteristic of an organism determined by genetic and environmental influences. You can see here on the picture on the top, genes, DNA. Genes contain instructions for making proteins. Here are the proteins. Proteins act alone or in complexes to perform many cellular functions. The other picture, homozygous alleles are same, heterozygous alleles are different, hemizygous, only one allele, for example, X or Y. Epigenetics. The study of how cells control gene activity without changing the DNA sequence. Epi means on or above in Greek, and epigenetic describes factors beyond the genetic code. Epigenetic changes can switch genes on or off and determine which proteins are transcribed. Here you can see an interesting picture about the epigenetic factor. You can see here the DNA, histone tails, chromosome, chromatin, and methyl group. Genomic imprinting. It is the process by which only one copy of a gene in an individual, either from their mother or their father, is expressed while the other copy is suppressed. Unlike genomic mutations that can affect the ability of inherited genes to be expressed, genomic imprinting does not affect the DNA sequence itself. Instead, gene expression is silenced by the epigenetic addition of chemical tags to the DNA during egg or sperm formation. Epigenetic tags on imprinted genes usually stay in place for the life of the individual. Angelman and Prader-Willi syndromes. Angelman, paternal allele silenced, and then the maternal allele, which normally would be active, is missing due to the deletion. For Prader-Willi, it's the converse. You can see here in the picture, Angelman on the right-hand side here, the paternal allele is silenced, and then the maternal allele is missing due to the deletion. On the contrary, Prader-Willi, in Prader-Willi syndrome, the maternal allele is silenced. It results in the missing part of the paternal allele. The paternal allele is missing due to the deletion. Angelman and Prader-Willi syndrome. Prader-Willi syndrome. The syndrome means learning difficulties, repetitive behaviors, hypotonia, and obesity. With the Angelman syndromes, then there is problem with the late or communicate poorly, epilepsy, generally have happy demeanor. Let's talk about epigenetic effects and behavior. Monozygotic, from a single fertilized egg, relates to identical twins. Dizygotic, non-identical or fraternal twins, where each twin derives from two separately fertilized eggs. Chromatin, part of the nucleus of a cell, consisting of DNA and histone proteins that makes up chromosomes. Transcription, the process of making a copy of genetic information stored in a DNA strand into a complementary strand of RNA. Finding genes linked to human behavior. Linkage analysis. It is a method that investigates if there is an association between identifiable pieces of DNA, genetic markers, and a trait of interest or disease. If the marker is present in all affected people, then a gene closer to the marker region is involved in the trait or disorder. Single nucleotide polymorphisms, or SNPs or SNPs. It's a variation in DNA sequence that occurs when a single nucleotide is altered. Genome-wide association studies is an examination of genetic variants to see if any variant is related to a trait. The method involves assessing if gene variations, called NSPs, occur more frequently in people with a particular disease than those without the disease. Quantitative trait locus, or QTL analysis. The statistical analysis and identification of DNA stretches linked to the genes with a particular trait. Transgenic, an organism that has a segment of foreign DNA incorporated into their genome. Translocation, transfer of a chromosomal segment to a new position. Neurodegeneration, Parkinson's disease. What? It's a brain disorder that causes unintended or uncontrollable movements, such as shaking, stiffness, and difficulty with balance and coordination. What are the symptoms? The first part is the motor function. tremor, muscular rigidity, bradykinesia, movement slowness, and hypokinesia, absence of kinesia. The second part is the non-motor function, lack of sleep, and depression. Parkinson's disease, pathology. There are two things here, loss of dopaminergic neurotransmission and depletion of other monoaminergic neurotransmitters in PD or in Parkinson's disease. The depletion of other monoaminergic neurotransmitters in Parkinson's disease altered serotonin and noradrenaline. Loss of dark staining in a small region of the midbrain, SNPC, or substantia nigra pars compacta. Component part of the basal ganglia, a collection of subcortical nuclei, important for the control movement. SNPC neurons look dark in humans as they contain pigment melanin, a byproduct of the synthesis of dopamine neurotransmitter. Gradual accumulation of cellular inclusions, Lewy bodies, composed of aggregates number of proteins, including alpha-synuclein. Parkinson's disease cause. There are three things here. The causes are neuronal death, genetic. Many of the genes identified involved in either mitochondrial function or the processing of unwanted cellular proteins. Environmental factors, exposure to toxins, pesticides, heavy metals, and infection, for example, influenza. There are three causes of Parkinson's disease: neuronal death, genetic, and environmental factors. What are the treatments for Parkinson's disease? Drug therapies and non-drug therapies. Drug therapies include levodopa. The side effects are dyskinesias or inability to control movements. The solution is to take another drug, the levodopa or dopa decarboxylase inhibitor combination. There are also non-drug therapies, for example, cell transplantation, deep brain stimulation, neuroprotective agents, and neuronal manipulation to increase dopamine production by using gene therapy and cell transplantation. Alzheimer's disease. Alzheimer's disease symptoms: poor memory of recent events. Over time, it spreads to inability to do normal tasks. Okay, let's see. Okay. What are the causes of Alzheimer's disease? Pathology and etiology. Pathology: neuronal loss, intracellular neurofibrillary tangles, aberrant aggregation of neuronal protein, tau, especially in the cerebral cortex and hippocampus in Alzheimer's disease, and amyloid-containing plaques, abnormal clusters of protein fragments that accumulate between neurons in Alzheimer's disease. Changes in neurotransmission. Extensive synaptic loss in many brain regions correlate with cognitive impairment. Reduced synapses results in the decline in neurotransmission level in acetylcholine and glutamate. Sorry for some of the typo errors here. Etiology: environmental factors, head injury, and genetics, OE gene. What are the treatments? Drug therapies, for example, cholinergic therapies and pathological modification. There are also non-drug therapies, for example, use mental activity. That's the end of this chapter. Hopefully you enjoy learning psychology with me. See you next time.